ముందుకు పెట్టి చెప్పరా నువ్వు ఆన్ అనగానే ఆపుతాను. Good evening to all of you. Welcome to Unacademy. Today our topic is Lac Operon Concept. Lac Operon Concept. So we'll discuss about this concept. What is Lac Operon Concept? Like that. Before starting about this concept, it's my I'm Anand Babu here from Vijayawada, Andhra Pradesh. I have teaching experience 15 years. My referral code Unacademy Babu Live. On my referral code you can get 10% discount. Okay. Here I would like to tell about the plus subscription here. Convenient plan is there for you. Okay, take subscription so that you'll be getting benefited. Okay, you'll be having regular interaction with educators and also separate batches for complete preparation, over which discussion will be there. So you'll be able to understand what is the concept clearly, and frequent tests and quizzes will be conducted for the convenience of you to make you perfect in preparing for examinations like EAMCET, NEET and other competitive examinations. Okay, on my referral code you can get 10% discount here. Okay, shall we start the program? Here, regulation of gene expression. What is Lac Operon? Lac Operon, what is it? Before knowing about the Lac Operon concept, we'll study about the regulation of gene expression here. What is the meaning of the regulation of gene expression here? Here you see, constitutive genes are those genes that are constantly here. Okay. Constantly here is the pen. Here I have to take this now. So here constitutive genes, there are two types of genes here. Okay. Sorry. Gene expression is different. Genes are expressed under different conditions. Okay, here constitutive genes here is here now. The genes are those genes that are constantly expressing themselves in a cell because of their products. Okay, are required for normal cellular activities. So for cellular activities, the genes will express. Here constitutive genes, two types of genes are there, constitutive genes and another one is non-constitutive genes here. So these genes continuously work, continuously function. Okay. For example, here if you take the glycolytic process, glycolysis is very important in which the glucose molecules get burnt, oxidized. Am I correct or not? So that energy is released. This energy is released in the form of ATP here. Okay, this ATP is very much required for cellular activities. Cellular activities here. Without this energy, cellular activities will not happen. I mean, organism cannot function. So here the genes control that process, glycolytic process, and also you can understand ATPase. ATPase. Here you see this is ATPase. How this ATPase is important for the formation of this ATP. This ATPase allow protons and due to the movement of the protons here, ATP energy is released. So the genes control this entire process, constantly works, but non-constitutive genes are there. They can be expressed only certain conditions. I mean, when there is a need, they can be expressed. When there is no need, they cannot be expressed. So the type of genes are called non-constitutive genes. Non-constitutive genes, here you see, are not always expressing themselves in a cell. These are called luxury genes. These are switched on and switched off according to the requirement. So according to the requirement, the genes can be expressed or may not be expressed. That point you have to remember. For example, if you want light, we will switch on. If you don't want light, we will switch off that. So like that, genes will be expressed in such manner. Okay. So we'll study about that now. So here, one more important. So here genes, some constantly work and control some of the processes which are important for cellular activities. Non-constitutive genes are those which work only under some conditions when there is a requirement. When there is no need, these genes will be switched off. These genes will be expressed or switched on only when there is a requirement here. So what is about that? Let us study now. Here you see operon. What is the meaning of the operon here? Before knowing that, so here this is operon. Operon means here nothing but these are all, what are they? Nucleotide sequences. I am particular genes here. Here you see this is a this is operon. Operon is nothing but, what is it? It is a structure consisting of different nucleotide sequence and DNA here. This is the promoter region and this is inhibitor region. One more promoter region here and here operator gene and ZYA structural genes are present here. But one point you have to understand here. What is the operon means? Consisting many genes. DNA consisting many genes. So this operon is very, very important. Okay. This operon concept is observed in E. coli. Escherichia coli bacteria, it is observed. Escherichia coli bacteria here. So when does gene express? When does a gene do not express here? So that is an important point. So the operon is nothing but DNA consisting the genes, polycystonic genes here, polycystonic DNA here. So this is a promoter, two promoter regions. This is inhibitor gene here, and ZYA are the structural genes. These are all structural genes here. So what is the importance of promoter gene here? Promoter gene is very important, with which RNA polymerase binds. RNA polymerase, RNA pol. RNA pol is nothing but enzyme which is very important for a transcription process. Okay, DNA to RNA transcription process. Okay, our RNA polymerase is there now. This RNA polymerase is very important for synthesis of mRNA and proteins like that. Okay, so this is RNA polymerase here. So promoter region is the here the unit with which RNA polymerase binds. Okay, so this RNA polymerase moves along with this operator gene, Z gene, Y gene and A gene like this, structural genes. But one point you have to remember, this operator gene allows this RNA polymerase all along the segment here, Z gene, Y gene, A gene, structural genes like this. Okay, then what happens, these are able to, these genes are there now. These genes are able to produce, what is it? mRNA. Synthesize mRNA and finally this mRNA and forms what? Proteins here. Here also mRNA like this and proteins. Here also you see Z gene and here proteins are formed. Proteins are nothing but enzymes here. Okay. So this is about the Lac Operon. Lac means lactose. Operon means DNA consisting different genes here. So this is the structure of the operon consisting promoter, operator, inhibitor and structural genes. What are the important functions of these? Let us study now. So here you see. So this is structural gene here. Lac Operon consists of the following structural genes. The genes code for proteins needed by the cell, which include enzymes or other proteins having structural functions. Okay, these are what this is A gene and Y gene. This is here you see. This is A gene. Sorry. So this is gene A. This is gene A. Sorry. This is gene A here. Okay. This is also called what is it? Transacetylase enzyme. This transacetylase enzyme function was not known. And next, Lac Operon Lac Y gene coding for enzyme permease. This is one enzyme permease that is synthesized by Y gene here. So, for example, you take the Y gene here. So, this is Y gene. Y gene is important and synthesis of mRNA. And finally, here the permease is formed. Permease. Okay, this is enzyme. So, Y gene synthesizes mRNA. mRNA forms, this is a enzyme that is what is called permease here. So, this Lac A gene here is there now. The function was not known here. And here operator gene here. Operator gene, it is nothing but a transcriptional structure. It interacts with the protein molecule, regulatory molecule which promotes. So, already operator gene. For example, here you see, this is operator gene. Here this is operator gene. So this operator gene allows the RNA polymerase all along the structural gene ZYA like this. But here one point you have to understand, this operator gene will function only promoter region is free. Promoter region is free. I mean promoter region should be, operator gene should open here. Should open. It should be switched on. Then only RNA polymerase move all along the this operon, ZYA operon. That point you have to understand. Okay. But what is the important function of inhibitory gene here? Inhibitory gene is also known as promoter gene is the gene where RNA polymerase binds. Inhibitory gene, this is also called regulatory gene. This synthesizes mRNA and produces one kind of protein. This is repressor protein. So this is very, very important here. For example, here bacteria is there. This is E. coli bacteria. Around the E. coli bacteria here, no lactose is there. Lactose is not there. Lactose is source of energy, no? It is absent. Whenever it is absent, what happens? This gene, the inhibitor gene produces what is it? mRNA. mRNA forms repressor protein. The repressor protein will be binding to the operator gene here. So, operator gene, what happens? Cannot function. It will not allow RNA polymerase to move along the ZYA genes. That is the important function of operator gene. Okay. So, this is the flow chart showing that functional state of Lac Operon in E. coli growing in the absence of lactose. Lactose is absent here. Whenever lactose is absent, if lactose is not present around this E. coli bacteria here, here the repressor protein binds to the operator gene. Operator gene does not function here, so that it will not allow RNA polymerase to move along with the ZYA genes. So here switch it off. I mean the expression of gene will not be functional, so it will be switched off. So Lac Operon concept that will not be expressed here. That point you have to remember. Another important point you see here. So this is the flow chart showing functional state of the Lac Operon in E. coli growing in the presence of lactose. For example, here you see this is E. coli bacteria, and it is surrounded by this lactose here. Lactose. That point you have to remember. Lactose is here. So in presence of lactose, what is about this Lac Operon? Lac Operon here is operator gene here will be free from this repressor protein. Operator gene is free here. That point you have to remember. How operator gene will be free from this repressor protein here? One point you have to remember. Here, whenever the lactose is present around this E. coli bacteria here, immediately here Z gene is there, no? The Z gene is able to produce, here is already beta galactosidase enzyme is secreted here. So it's inside. First transcription takes place. I mean DNA to mRNA here. mRNA to beta galactosidase enzyme synthesis happens here. So whenever these beta galactosidase important function is beta galactosidase important function is what? Conversion of this lactose into glucose and fructose. Glucose and fructose here. Why like this? Glucose and fructose here like this. Glucose and fructose here. That point you have to remember. So whenever these beta galactose converts this lactose into glucose and sucrose, what happens? Tell me here. Here one more important point you have to understand here. So the glucose is absorbed by the E. coli bacteria. Glucose is absorbed by the E. coli bacteria. Here one more important point you have to understand here. Okay. So whenever this is formed, lactose is formed here. So this lactose is lactose. The lactose binding to repressor protein here. This is repressor protein with which this is binding here. So that repressor protein will be inactive. So it will not function. So now operator gene will be free. And it allows what is the RNA polymerase from promoter origin here. So the RNA polymerase move all along the structural genes here. And then what happens? The Z gene here producing what is it? Beta galactosidase enzyme by translation. First transcription followed by translation here. In transcription, synthesis of mRNA happens. This mRNA secretes synthesize the beta galactosidase enzyme. Whenever beta galactosidase enzyme is synthesized, what happens? The lactose that is present around this E. coli is converted into what is that? Glucose and fructose. That point you have to understand. So here this glucose is absorbed into the E. coli by one more enzyme here. What is the enzyme here? Permease. The permease enzyme here is secreted. Here the permease enzyme is secreted by what? Y gene. Y gene here is producing what is it? Permease. This permease enzyme is generally present here in the membrane of the E. coli here. Okay, through which the glucose here enters. Glucose enters. Glucose is source of energy. In this way, the E. coli bacteria will be getting energy here. But E. coli bacteria getting energy only when it is surrounded by what is it? Lactose. If lactose is not present, what happens? Gene expression will not be there. I mean the repressor protein will be binding to the operator gene here. So that what happens? This, what is that? Absorption of glucose will not happen. Beta galactose will not produce this, this Z gene will not produce this beta galactosidase. That point you have to understand here. So this is the flow chart representing the, what is that? Representing functional state of the Lac Operon in E. coli growing in the presence of lactose as a sole carbon source. I mean glucose is a carbon source over here. So when E. coli is surrounded by these lactose, surrounded lactose, then only gene expression will be there. How gene expression is controlled? By repressor protein here. Repressor protein positively, negatively works. If lactose is not present around this E. coli bacteria, what is about the gene expression here? Here is the gene expression. So immediately here what happens? And this inhibitor gene here, no, this synthesizes mRNA transcription and synthesizes the repressor protein. This repressor protein binding to the operator gene. So operator gene will not, it will be switched off here. So it will not allow the RNA polymerase here from promoter region to structural genes here like this. So that Z gene here is there now. The Z gene is unable to transcribe this enzyme, transcribe and unable to produce or synthesize beta galactosidase enzyme. So that what happens is it will be switched off. Lac Operon will be switched off. So it will not function. Am I correct or not? Whereas in case of this is when E. coli is surrounded by lactose. So the functional operon here you see what happens here. The inhibitor gene synthesizes this mRNA from which repressor protein is formed here, but already here lactose is there around the E. coli bacteria. The lactose binds to the repressor protein here. Here it will bind. So that what happens, tell me, the repressor protein becomes inactive. Whenever it becomes inactive, it will be separated from operator gene. So it will be separated from operator gene here. Okay. And finally what happens here, the promoter gene, this operator gene allows that type of RNA polymerase, and RNA polymerase move all along this structural gene ZYA. Here already I told you A is gene synthesizing what is the transcription, synthesizing what is the translation process is. Okay. First this gene synthesizing what is it? mRNA here. That is transcription, DNA to mRNA transcription. And mRNA to this enzyme, that is a protein. That is what is called translation here. So enzyme that is synthesized by A gene here, transacetylase, its function was not known clearly here. But Y gene here is there now. This Y gene is able to synthesize this permease enzyme. In what way permease enzyme is important? This enzyme is present in the membrane of the cytoplasmic membrane here, like this in E. coli. So this permease enzyme allows the glucose from outside to inside. So glucose is a carbon source of energy here. So this energy will be utilized by the E. coli bacteria. Glucose is, this is what is called lactose. Lactose will be in the form of glucose. Lactose will be in the form of glucose. Already I told you know, beta galactosidase enzyme that will act on lactose and converts it into glucose and fructose here. So in the form of glucose, E. coli bacteria absorbs this lactose inside. So this is Operon concept. What is the important point here? I will tell you. So Operon, Jacob and Monod. Here is Jacob and Monod. Monod and here Jacob. Proposed a model of gene regulation known as the Operon model. Operon is a coordinated group of genes. I mean DNA consisting the genes which are in coordinate function. I mean they coordinate each other. I mean there will be coordination among the genes such as structural gene, operator gene, promoter gene and regulator gene. All these genes work together. Am I correct or not? That is nothing but a coordination among these genes which function or transcribe together and regulate the metabolic pathway. Metabolic pathway means what is it? When E. coli is surrounded by lactose here, in the form of glucose, energy is obtained by it. When E. coli is not surrounded by this lactose, so gene expression will not be there. Okay, it will be switched off. Switched on mechanism here. Operon, this is the definition for Operon. And who proposed this Operon concept in E. coli? Jacob and Monod. And also here regulation of genes here. And here in regulation of genes, there are two types of genes here, consecutive genes and non-consecutive genes. Consecutive genes here control the process like glycolysis, in which substrate molecules get oxidized and release energy. Continuous production of ATP is very, very important. So continuously the genes gene products function. Non-consecutive genes, these non-consecutive genes do not work continuously. When there is requirement, they will be expressed. When there is no requirement, they will not be expressed. These genes are called luxury genes also. Okay. So, for cellular activities here, for example, genes are nitrate reductase in plants and lactose system in E. coli. Lactose here. We studied here, this is the lactose. Our topic is. Okay. Maximum functional efficiency to the cell. Such regulated genes therefore are required to be switched on, switched off when particular function is to begin or stop. This regulation can be achieved by any one of the following is induction and repression. So induction means what? This is a process of gene regulation where the addition of substrate stimulates. Substrate means here what is it? Tell me. That is the glucose. Stimulates the synthesis of enzymes. What are the enzymes needed here? Beta galactosidase and also one more enzyme, that is permease, and also trans-acetylase enzyme. Okay. Repression means inhibitory gene is there, no? So the addition of this end product stops the synthesis of enzymes. When do you find the non-functioning of the enzymes? Whenever repressor protein binds to operator gene. So that will happen only when E. coli is not surrounded by lactose. Am I correct or not? So about the regulation of genes, this is information. Okay, operon, what is the definition for operon here? This is the operon definition. What are the elements present in operon here? Operon is nothing but a DNA consisting different types of genes which work together and maintain the entire process, switched on, switched off mechanism. Okay, and here you see what is about the structural gene and everything I will tell you. So this is the flow chart showing when E. coli is not surrounded by any lactose here. Lactose is not surrounded, E. coli not surrounded by here lactose. So in absence of lactose, there is no need of getting energy here. Lactose is absent now here. So this entire process will be switched off. I mean gene expression will not be there. So here already I told you promoter region is the region with which RNA polymerase binds. Okay, and inhibitor gene is here. This synthesize mRNA by transcription, and mRNA by translation forms this repressor protein here. The repressor protein acts negatively and positively. When does it act negatively here? Whenever this is not surrounded, E. coli not surrounded by this lactose here, this repressor protein binds to operator gene. So operator gene will not allow what is the RNA polymerase from promoter region towards structural genes here like this. Okay, so that what happens, the Z gene will not produce synthesize the beta galactosidase enzyme. Okay, okay. So it will be switched off. Gene expression will not be there now. So Lac Operon will be switched off. And Y gene here is there. The Y gene is important for permease, secreting permease. By transcription, here DNA, mRNA synthesis, transcription takes place by transcription, and by translation, mRNA synthesizes that permease. But permease is not formed here. Why? Because the structural genes will not function here. What is the reason here? Operator gene is not allowing. Operator gene is switched off here. It is not permitting the RNA polymerase from promoter region here. So the structural genes will not function, so that particular kind of enzymes are not synthesized. The entire process will be switched off here. So this happens only E. coli is not surrounded by the lactose. That point you have to remember. And also here you see, when will it be expressed? Operon will be expressed whenever this operator gene is free from this repressor protein. When does this repressor protein from operator gene is free? Okay, when E. coli is surrounded by this lactose. When it is surrounded by lactose, what happens here? You see, the lactose here is there now. It binds to repressor protein here. So immediately what happens in repressor protein? Chemical activity will be changed. Some conformational changes will happen, so that it becomes inactive. I mean, it leaves the operator gene. It will not function now. Repressor protein. Whenever repressor protein is free from operator gene, immediately what happens here? You see, the promoter gene RNA polymerase binds to promoter region. The RNA polymerase, you see, addition of nucleotides for the purpose of transcription, mRNA synthesis, it moves, and the operator gene will open, and allows the RNA polymerase here towards the structural gene ZYA here. ZYA now will be functional. Z gene here by transcription synthesizing mRNA. Okay, Z gene here, and from mRNA beta galactosidase enzyme is synthesized. This is important in the conversion of lactose to glucose and fructose. Glucose source of energy. This is absorbed by the E. coli by permease enzyme. The permease enzyme is synthesized by Y gene here. Okay, Y gene starts to synthesize this permease enzyme first by transcription and followed by translation here. Permease enzyme is secreted here, synthesized. Permease enzyme is present here only, in the cytoplasmic membrane here, through which the glucose is absorbed. Actually here one more important function. Inside the cytoplasm directly glucose is not there. Inside this E. coli bacterium here, the glucose, here some lactose is there or glucose or one more amylopectin. So, okay, in the cytoplasm this will happen. Glucose is absorbed, and finally what happens is the repressor protein becomes inactive because of the changes in the chemical activity in the repressor protein here. So, Operon concept will be switched on. Operon concept will be switched on. Okay. So here today the pen is not allowed to write properly. Sorry for the inconvenience. So I'll make it correct next class onwards. So regulation of gene expression. What is the overall meaning of the regulation of gene expression? Genes are expressed differently. Okay. Some of the genes constantly expressed, they are called constitutive genes. Okay. For example, to control the glycolysis and ATPase enzyme here, the constitutive genes are there. Constitutive genes are very important, which continuously work. What is the reason? This process should take place for cellular activities continuously here. Another type of gene, this is non-consecutive genes. They work only when there is a requirement here. When there is no requirement, requirement here means, example I told you, when E. coli is surrounded by lactose only. If E. coli not surrounded by the lactose here, okay, non-consecutive genes will not be expressed. That point you have to remember. That is nothing but a gene expression here. So, and genes are switched off and switch on depending on the need and depending on depending on the need, okay, or based on the requirement here. So here one more important point here. What is the operon? Operon is nothing but a group of genes which are in coordinate function here, all work together and see the process of pathway, that is expression of gene or not expression of the genes here. Switched off, switched on mechanism here. So this Operon concept was studied clearly by Jacob and Monod here. And here you see different types of genes which are present. Operon here, structural genes. Structural genes are nothing but what are they here already explained. Promoter genes. Actually promoters, two promoters will be there here. Upstream of the operator gene, this promoter gene will be there. This is inhibitory gene. This is also called regulatory gene here. Promoter gene is a gene with which RNA polymerase binds. Operator gene allows the RNA polymerase from promoter only when it is free from repressor protein. Okay. That point you have to understand. Here this is the diagram showing only when E. coli is not surrounded by this lactose. So structural genes will not be functional here. An entire operator, that is Lac Operon concept will not be expressed here. Okay, that's fine. And this is the diagram showing what is the gene expression here. ZYA genes will be functional here. What is the reason means here? Operator gene is not binding to this repressor protein here. How repressor protein becomes inactive means when it is associated with the lactose here. So lactose is present around the E. coli. This lactose binds to this repressor, and repressor protein becomes inactive. So it will leave the operator gene. It becomes non-functional here. Whenever it is non-functional, immediately what happens? Operator gene allows RNA polymerase from promoter region and this RNA polymerase moves all along the ZYA genes, structural genes, and promote the synthesis of enzymes. From Z gene, beta-galactosidase, from Y gene here, permease, and A gene, trans-acetylase. So these enzymes are released only when operon is switched on. This happens only when E. coli is surrounded by the lactose here. Important function that is concerned with the beta-galactosidase, which is synthesized by Z gene here, and it will convert lactose into glucose and fructose. Glucose is a carbon source. Okay, so this glucose is absorbed by the E. coli when it is surrounded by glucose, when this operon concept is switched on. Okay, so this is the way how this E. coli bacteria utilizing that lactose. Okay, this is a switched off, switched on mechanism. These are very important concept. This is so in the next lesson we will come to we'll come with another topic here. Okay, here you see, I will tell you. On the other hand, you see here, when inducer means here lactose only. Inducer here is nothing but a lactose here. Lactose. Okay, whenever lactose is formed here, it binds to repressor protein. Repressor protein by gene gets bounded to it. The repressor therefore gets released from the operator gene. RNA polymerase is now permitted to act. Hence, the transcription of lac genes now takes place and the operon model in switched on position. All three genes are transcribed by the RNA polymerase to form single mRNA strands here. So, okay, thank you very much for listening here. In this lesson, you have studied that what is regulation of genes, how genes will be expressed, okay, under different conditions here. What are consecutive genes, non-consecutive genes, what is the meaning of the operon here, and who proposed this operon concept, Lac Operon. Lac means here lactose. Okay, what are the components present in DNA? Okay, structural genes, Lac A gene and Lac Y gene, and also Lac Z gene. The three genes are functional, having different functions here. And operator gene, it is very important gene with which repressor protein here binds. So structural genes will not function here. Promoter gene is a gene with which RNA polymerase binds here. Okay. Regulatory gene, it is also called inhibitory gene. Inhibitory gene by transcription synthesize mRNA, and from mRNA this what is that? Repressor protein is synthesized here. So this is allosteric protein which can exist in two forms. In one form, it paratactic and operator binds. Operator binds. On the other hand, it what is it? Paratactic to the inducer, such as lactose here. Okay, do you understand? Thank you very much.